Okay, we'll wait for 2 more minutes and then we'll start with the class. Okay, so we will start with today's class. It's already 7:10. Other people can keep joining. So today we are in the week 9 already. So we have already discussed 8 weeks of live sessions for the course Industrial Biotechnology, and today we will be discussing week 9. So today I have a set of 12 questions, 12 MCQs, which we will be discussing in the next 1 hour. And the pattern is same. You have to um, you will be shown the question, then you have to think for its correct answer for around 2 to 3 minutes, and you have to write down your correct choice of answer in the chat box. And then after some time we will discuss what is the correct answer and what is the correct explanation to that correct answer. Okay, so this is the pattern we have been following for several weeks now, and we'll stick to that. And other than that, uh, everyone knows the rules for the class. Uh, we are Please keep yourselves mute for the audio and video. And I'm having a little bit of cold, so I may not be uh, that much loud. So if you're unable to listen properly, then let me know. Okay. And if you have any kind of doubts for the last 8 weeks of interactive sessions, or today, anywhere, any questions, if you're having doubts in the questions we are discussing in the live sessions, or the questions you all are answering in the current assignments, anywhere, if you have any doubt, you can ask it here and I will clear it for you. And I have been doing this for some of you in the last classes, and this I can continue. If you have any doubts from the current year assignments, or if you think that those are someone mistakenly they have given wrong answers, then we can discuss it here and we can know what is the right answer to it. Okay. I asked Majid, you have written pardon me, ma'am. Can you tell me what's the doubt? Like, write it down in the chat box if you have any doubts. Okay. Other than that, we'll start with today's class. It's already 7:13. So I hope everyone is clear. We'll start with the first question of today. Which of the following is not a desirable characteristic of Baker's yeast used in fermentation? High fermenting power, genetic stability, resistance to autolysis, requirement of expensive growth substrates. Okay, start answering. Okay, let's see what is the correct answer to this question. It is D. Requirement of expensive growth substrates. So Baker's yeast is selected for traits like genetic stability, high fermenting power, rapid growth on cheap raw materials, and resistance to autolysis. Using expensive substrates would reduce cost effectiveness and is undesirable. Okay, so I hope this is clear. So requirement of expensive growth substrates is not needed for Baker's yeast. Uh, is not a characteristic of Baker's yeast which is used in the fermentation. Okay, we'll move on to the next question. In Baker's yeast fermentation, agitation of the medium is avoided because it reduces oxygen availability, it causes foaming, it leads to cell lysis, it lowers pH of the medium. That's great. Okay, I hope my voice is audible, right? Yeah. Okay, let's see what is the correct answer to this question. It is C. It leads to cell lysis. Although aeration is essential, direct agitation can rupture fragile yeast cells. Hence, air is supplied uniformly without strong agitation to maintain oxygen contact while preventing lysis. So, in Baker's yeast fermentation, agitation of the medium, like shaking of the medium, is avoided because it can lead to rupture of the yeast cells. Aeration is important, like the area uh, kept empty for the air to pass is very important, and hence air is supplied uniformly without strong agitation to maintain oxygen contact while preventing lysis. So, agitation is avoided because it can cause the rupture of the cells, or you can say it can lead to cell lysis. That's what it is. Moving on to the next question, third question: which of the following yeast species is most commonly used for fodder yeast production? Saccharomyces cerevisiae, Candida utilis, Cuveromyces lactis, Hansenula polymorpha. Start answering. Think properly and answer. Okay. Let's see what is the correct answer to this question. It is Candida utilis. So, Candida utilis, which is also known as the Torula yeast, is widely used for fodder yeast because it can utilize both hexoses and pentoses from waste sulfite liquor, has high protein content, and provides vitamins of the B complex. So this is why the Candida utilis, which is the B option, is used for fodder yeast production because it can utilize both hexoses and pentoses from waste sulfite liquor. So it can utilize the carbohydrates from the sulfite liquor, has high protein content, and provides vitamins of the B complex. That's why this one is used for fodder yeast production. Moving on to the next question, the main industrial advantage of Candida utilis over Saccharomyces cerevisiae for fodder yeast production is higher ethanol production, ability to use pentoses, better tolerance to acidic pH, resistance to molds. Yeah, this is a easy one because already we discussed the third question. Still, I'll give you 2 minutes time. Okay, let's see what is the correct answer to this question. It is B, ability to utilize pentoses. As we discussed, Saccharomyces cerevisiae can only utilize hexoses and disaccharides but not pentoses, whereas Candida utilis can utilize pentoses present in the waste sulfite laker, making it more versatile for fodder yeast production. Right? Moving on to the next question, spirulina is classified as a green algae, chlorophyta, a cyanobacterium, a red algae, rhodophyta, a diatom. Start answering. Okay, let's see what is the correct answer to this question. It is A, cyanobacterium. So spirulina belongs to cyanobacteria, which is called as the blue-green algae, specifically Arthrospira plantaris and Arthrospira maxima. These are the ones which are classified as the blue-green algae. It is a filamentous prokaryote used as a food and feed supplement. Okay, so you all must have heard about spirulina, that generally it is also taken as a food supplement because of its nutrition content. Okay. Moving on to the next question. Which of the following conditions is maintained to prevent contamination during large scale spirulina cultivation? Neutral pH, high temperature, that is more than 40 degrees, high alkalinity, pH is greater than 9, or low illumination. So, which of the conditions is maintained to prevent contamination during large scale spirulina cultivation? Start answering. Okay, let's see what is the correct answer. It is C. High alkalinity, that is pH is greater than 9. If the pH is less than 7, it is acidic, and 7 to 8 is considered as the neutral pH, right? This is a basic thing, we all know it. So, maintaining pH above 9 is critical because it suppresses contamination by other algae and microbes, while spirulina can thrive under alkaline conditions. So, spirulina is known to what we say thrive in the uh, alkaline conditions, that is the basic conditions, that is pH is greater than 8 or 9, whereas other algae and microbes, although spirulina is also algae, but it is known to maintain itself in alkaline conditions as well, whereas other algae and microbes, they can't do it. That's why that conditions is used to prevent contamination for large scale spirulina cultivation. Okay. Moving on to the next question. The primary advantage of spray drying in spirulina processing is it is the cheapest drying method, it preserves heat sensitive nutrients, it produces higher protein content, it reduces contamination by fungi. Start answering. Okay, let's see what is the correct answer. It is B. It prevents heat sensitive nutrients. So, spray drying ensures rapid drying that preserves pigments, enzymes, and nutrients, making it superior for large scale production despite higher cost compared to sun drying. So, spray drying is preferred for spirulina processing because it preserves the heat sensitive nutrients which can be lost if we are using the sun drying method. Okay. Moving on to the next question. Which type of amylase cleaves internal alpha 1 4 glycosidic bonds randomly within starch? Alpha amylase, beta amylase, gamma amylase, glucoamylase. Start answering. Yeah, whatever questions you have, please ask it in the chat box. I will see. Okay, let's see what is the correct answer to this question. It is A, alpha amylase. So, alpha amylase is an endoamylase that cleaves alpha 1,4 bonds within amylose and amylopectin, producing dextrins and oligosaccharides. And beta amylase works exovise from the non reducing ends, while gamma amylase acts on the alpha 1,6 and terminal alpha 1,4 bonds. So, the question was, which type of amylase cleaves internal alpha 1,4 glycosidic bonds randomly within starch? So that is alpha amylase, right? Moving on to the next question, which microbial group is most commonly exploited for large scale industrial alpha amylase production? Plant seeds, animal pancreas, bacteria, bacillus species, or cyanobacteria? Start answering. Since this course is offered from the last 8 years, from where can we get the previous year questions? Uh, Hi Vartika, but I think you can't get the previous year questions because they didn't even give me the previous year questions. Actually, till last year, I was uh, taking live interactive session for other courses like animal physiology and everything. And there I was discussing the previous year questions, assignment questions, which were given to me. But currently, for this course, they told me that I have to prepare my own set of questions and they won't be giving me any previous year questions. So I think all don't have access to those previous year questions. It's not possible, I guess. Yeah. Otherwise, if they would have given me the previous year questions, I would have only discussed with you all. But they told me to make my own questions. They didn't give me the previous year set of questions. Otherwise, till last year for the other courses which I was taking, I was been given the previous year questions by the NPTEL team. Understood? You can search on internet if in anywhere that you have to search and see. I search once, then let me know if you are able to find it or not. As far as I know, I don't think that there. Just try it once, okay? That I don't know, Vartika. As I told, I have not been given information about the exam. I think it's written on the NPTEL website that how many marks of exams it will be and what is the pattern of exams. I think it is written. Please go to the NPTEL website for this course and check there once. Okay? I don't think it will be only MCQs. I am not sure about it. You have to check it. Okay? Those things, but what will come in the exam and all, that is decided by the course lecturer itself. Okay, it's only mentioned about marks. As I said, I am not the in charge for preparing the questions or giving you marks, anything. Everything is done by the uh, course instructor. Okay. So if you really want to know, mail to the NPTEL team once. Maybe they can help you with this. I can't ask because I'm not assigned for that thing. You all can mail, but if they are not replying to you, then I can mail saying that okay, they are not replying to your mails. Okay. Okay, then I will mail. Okay. Let's see, I will mail. And if they reply, they should reply to me at least. If they know, then maybe they will reply. Or else they will say something. But if the exam pattern is completely on the instructor, just have to wait. Right? Let's see. I will try to talk to someone and get back to you next week. Okay. Okay, let's see what is the correct answer to this question. It is C. Bacteria, which is the Bacillus species. So, Bacillus subtilis, Bacillus licheniformis, and Bacillus amyloliquefaciens are the major industrial producers due to rapid growth, extracellular enzyme secretion, and ease of fermentation. So, these are the ones which are commonly exploited for large scale industrial alpha amylase production, and fungal amylases, that is asparaginus, are also used, but bacterial sources dominate industrially. Fungus is also used, but mostly bacteria is used. Right? On to the next question. High fructose corn syrup is produced by converting glucose to fructose using glucoamylase, xylose isomerase, that is glucose isomerase, alpha amylase, or lactase. Start answering. Is it told that the question is the exam is computer based? Is it given that you will be having a computer based exam? Okay, then I think they will do it accordingly. Obviously, they won't give you questions which you can't solve it on the like in the computer, right? So maybe you won't have the graph based questions. If they are saying computer based, then maybe high probability is that you will be having only MCQs. Let me confirm there, or let me check myself once what is written for this exam pattern for this year. Okay, then I'll talk to you. But it is computer based, you should mostly, yeah, because those lecture materials are old actually. I don't think they are recently prepared. They are prepared long back and they have been kept in the website like that. So those are not the something that is prepared recently, right? Yeah, that's why. But the exam pattern keeps changing. When I also gave this NPTEL exam in my masters. That time it was paper based. We wrote in paper answers, MCQs and subjective both were there when I gave MSc exam in 2020. But it's been 5 years now, so they have made it computer based now, but they have not updated the lecture materials. So, but the question pattern may be different. Then it will be MCQs. Let's see. I'll talk. Okay, let's see what is the correct answer to this question. It is B. Xylose isomerase, that is glucose isomerase. So, Uh, high fructose corn syrup production involves alpha amylase to liquefy starch, glucoamylase to release glucose, and finally glucoisomerase, also called xylose isomerase, to convert glucose into fructose. So this is done by the glucose isomerase. Next question. Which of the following properties makes HFCS particularly attractive to food manufacturers? It crystallizes easily, it increases microbial spoilage, it is more hygroscopic than sucrose, it is more expensive than sucrose. Start answering. Yeah, if it is computer based, maybe in computer only they will allow calculator to you. Don't worry about those instructions. When your exams will come, they will give you all the instructions clearly, and then only you will be going to the exam. So don't need to worry about small things. Okay, they will be sorted. First of all, let me confirm if you have at all subjective questions, because it is computer based, so it is different. It is difficult attend subjective questions in a computer setup, right? So I excuse. Let's see. As I said, Vartika, I am not aware of the exam pattern, right? Like that part is not for me at all. Like I am not at all involved in the exam discussions. I only works to so if you want, please make a list of all the questions and email me so that I can send it to the NPTEL team. Better they can answer it. If I want also, I can't answer these questions which you are asking. Please make a list of the questions you want to ask to the NPTEL team. If you can mail, then fine. Or if you want me to mail, then this is the email ID I am giving you now. Please mail me your questions. I will forward it to the NPTEL team. Okay, but there is no point in asking me because I don't know anything. That is completely dependent on the course instructor or the one who has made the lectures, right? Okay, let's see what is the correct answer to this question. It is more hygroscopic than sucrose. So, this high fructose corn syrup has which of the following properties? So, it is more hygroscopic, that is retains moisture. Hygroscopic means what? It can retain more moisture than sucrose, has high osmotic pressure also, and prevents crystallization. These features enhances texture, moisture control, and shelf life of bakery products. It is also cheaper than sucrose, right? Moving on to the next question. The main reason HFCS replaced sucrose in the US food industry is superior nutritional value, lower production cost, and easier handling. Higher sweetness than sucrose, it contains no calories. Start answering. Ayas is the only one answering all the questions. I can see that. As I said, if it is only MCQs, the questions we are discussing in the class, live interactive class, is very important because these are the questions I am submitting to the NPTEL team as well. And they may consider these questions as well for the exam. You don't know it, right? So you have to practice everything which is given to you. So don't take lenience on anything and be prepared whatever material you have for the best, right? And all the viewers who watch on the YouTube as well later and have not joined the live sessions, for them also I'm saying very clearly that whatever questions we discuss in the live sessions are very important, and whatever current year assignment questions you're solving on the portal, that is also important as well. Okay. Okay, Vartika, if you have not attended the lecture, that is fine, but keep up with the pace which we are going on. The live sessions, we have only 3 more weeks left, and yeah, try to come prepared in the next class if not today, no problem. Okay, so this is the last question for today. Let's see what is the correct answer to it. It is B, lower production cost and easier handling. So HFCS became popular because it is cheaper than sucrose, liquid easier to transport and blend, and extends product freshness. Its adoption was mainly economic, not nutritional. So the main reason why this corn syrup replaced sucrose in the US food industry is because of its low production cost and easier handling, because it is liquid in nature and it is also cheaper and it is economical, right? So this was the last question of today's live session. Last one. Okay. Uh, I gave this NPTEL exam in 2019 or 2019, I think it was. Uh, I won't say it was easy, but I won't say it was tough as well. The question paper level was moderate, and there were MCQs and subjective type both. And MCQ, that time these live interactive sessions were also not there, because this started because of PMRF. The people, the students who, the PhD students who got PMRF, they are taking these live sessions. So PMRF was not there that time. So live interactive sessions was also not there. So whatever MCQs we got, it was all from the current year assignment questions we have done, and we had done, and subjective questions were also there, like for 5 marks, like that. But I had taken some other groups. It was not industrial biotechnology as far as I remember. It was some, I think, techniques wala course I took some. I remember like microscopy, spectroscopy, all those were in that thing. So yeah, it won't be too difficult, but it won't be very easy also. It will be moderate. If you have read, obviously you will feel it's not tough. If you have not read, you will feel it is tough, right? It all depends on you how much you are prepared. Okay, just mail me the set of questions you want. I will forward it to the NPTEL team. Don't keep it much lengthy. Write it in a crisp manner, whatever major doubts you have. Okay? Otherwise, if I send a very big mail, I don't know they will answer or not. Okay. Okay. So I asked how many marks, how many correct answers have you given out of 12? Let me know if you want to share your score. You had it to get it out scores. So, okay. So if you all have no doubts, we can disperse. Again, we will meet on next Tuesday at 7 o'clock. Let's try to be on time. Okay. Thank you, everyone.